Thank you very much. It's great to be here in Estonia. It's only my second visit to Estonia and my first to wonderful Tartu, so thank you very much for having me. Why am I here? The first is to add to Anders' recommendation of thinking about happiness and the Happiness Foundation. Secondly, I'm discussing collaborations in Estonia with a company that I've set up to apply artificial intelligence to medical clinical decision-making, and this is a place with great data and very enthusiastic doctors and technologists. But the third is to share with you some secrets of successful innovation that come from quite a long career, as you can see from the color of my hair. So I'm difficult to pigeonhole because I have spent time in academia, I've spent time in industry, I've worked in startups, I worked in large companies, I've worked in the National Health Service. So in a way, I've both been part of originating innovation, developing innovation, and also consuming innovation. So it's important you know a little bit of where I'm coming from. So here are the secrets. Some of them are rather obvious, I have to admit. Work back from the problem, engage with users, evaluate things in parallel. I'll explain what that means in a minute. Challenge conventional wisdom, obviously. Plan evidence collection from the start, probably also obviously. But then I'll slow down when I get to 6, 7, 8, 9, and 10, which is about understanding where the gap is between the innovation we might have and the innovation we actually see in daily use. And I'll talk a little bit about precision innovation. And those two things are the subject of a book I bought a copy of, and if anybody wants it from the bioscience community, feel free to take it away. Early conversations and partnerships with people who might eventually acquire what you are. And then 9, let's see if we can distill some of the lessons from the COVID pandemic. And then I'm going to end up with some rather radical ideas, which hopefully will spark some discussion, how we might challenge the overall European innovation model, which I would argue, with some bright exceptions like Estonia, actually isn't working very well. The first thing is how to start. So I'm a scientist and technologist. So I start firmly on the left-hand side, and I work left to right. If you've seen these charts, these chevrons that march inexorably from left to right, I start from left to right. I ask myself the question, this technology, is it cool? Can we patent it? Will investors like it? And how quickly can we bring it to market? Well, those are not stupid questions. I mean, they're reasonable questions. But actually, truly successful innovators start the other way. They start from right and move to left. Who has the problem? Where do these problems exist? And therefore, who will be the early adopters of my innovation? Who's going to see immediately they want to use this? How do we, in fact, make it attractive to them? And can we turn them into champions of that innovation? So work from right to left as well as left to right, depending on whether you're, in fact, a commercial person or like me, a scientist and technologist. Secondly, engage a variety of users. This doesn't come up quite the way it should, but never mind. What it should have is a depiction of the wide variety of users that you need if you're, for example, innovating in the healthcare space. You have to worry about doctors, nurses, health managers, patients, most importantly. And think about the perspective that all of them will have on your innovation as it both is developed and comes into use. Don't just think about the most obvious customer. The third is evaluate in parallel, not in series. What goes with this idea of going from left to right, or as you see it, that way, is you tend to evaluate the technology first and solve your technological problems. Then you pause and say, is there going to be a market for this? And so you do market research, you have discussions with users, and you try and figure that out. And then ultimately you say, well, okay, what about the economics of this innovation? How will it cost me? How much will it cost me to make? What can I sell it for? In other words, what's the value in the hands of the consumer? And is there a profit margin? It's pretty obvious once you say it that you need to evaluate those things in parallel. You can waste a great deal of time and money if you just evaluate the technology, move to marketing, and then do the economics. Let me give you one example from my domain, bioscience. There's a wonderful, wonderful technology called CAR-T therapy. Anybody heard of CAR-T therapy? It would help me if there was one hand up. There isn't even one hand. There's one hand up. Wonderful. So CAR-T therapy, you will have read about it in the newspapers. You may not have heard it called that. You engineer the T cells from a patient to recognize and kill particular tumors that they have. You have to take the cells from the patient. You take them to a lab, sometimes down the street, sometimes hundreds of miles away. You engineer them, and then you take them back and re-inject them into the patient. Very, very expensive. Same is true for any kind of gene therapy, actually. There's one on the wires today in the UK that will cost 1.5 million per patient. So pretty obviously this is expensive. Pretty obviously the market is rather small. But actually the people who are innovating, and I was in a company that was doing just that, didn't think about the economics from the point of view of the hospital. Turned out that even if you could persuade the reimbursement authorities, which wasn't easy, to reimburse it, the hospital lost money on every patient they treated. Not a good idea. So think about all of those three topics in parallel. Then be unconventional in the way that you think about things. Apologies again that this is a bioscience example, but how are drugs normally developed? Well, we have something called randomized controlled trials, phase 1, phase 2, phase 3. In these, we test on a pretty large number of patients over a pretty long period of time before we get to the magic moment, as it's called, the moment in which the drug is approved. After that, there's very little serious study. You see the blue area, patients are treated with no active surveillance, it says. There are a couple of consequences. It takes a very long time and a lot of money to get to the magic moment. And then actually, there's a lot of learning after the magic moment that is never captured. So with some other folks in Europe, I innovated something called the adaptive pathway. So what you do is you have two moments. You do some smaller trials to figure out, is there actually a case for this? Does this work pretty well? Maybe we could have tried it in thousands more patients, but does it work? Is it safe and effective to certain standards? And you give it an initial or conditional license. Then you do an increased number of patients that are actively surveyed, and you can expand to a larger profile of patients. And so there are less patients actually this way treated without active surveillance. And you can give something a full license at a later point. And the overall cost and time associated with this is much less. Collect evidence as soon as you can and as often as you can, but also from the point of view of the different users that I mentioned before. Doctors, patients, payers, regulators, health technology assessment agencies. Don't get to the end, to your either single magic moment or second magic moment, and then say, oh, how do I prove the value of this drug? You've got to be collecting that evidence in the course of the trials. Sorry, I skipped one because I didn't expect to see it. As well as the cost associated with bioscience development, there's a huge attrition rate. Let me point out that this is an exponential or logarithmic scale. Every year, or at least the year in which this analysis was done, I did this analysis, there were 2 million papers published in bioscience. Of those, you could say maybe 300,000 were, first of all, repeatable, but secondly, useful and relevant to the treatment of human disease. And there were 14,442 patents registered that year, and 5,400 drugs in the pharmaceutical pipeline. But out of all of that, only 34 drugs were actually approved. So this is a terrible fallout rate. And the other problem is the time. 20 years to go from the recognition, well, actually, as you can see here, 50 years from identifying the chromosome associated with chronic myeloid leukemia, all the way through to being able to clone it, then discovering the first designer drug in cancer, which is called Gleevec, and then the FDA approval, before you get to the point of blockbuster sales at $4.7 billion in the 2010s. It's a hugely lengthy process, hence the need to rethink. I won't take time on this, but I've analyzed, and again, you could read about it if you like. It's not just one development gap. There are many gaps that exist in the development of drugs and bioscience products. The point of putting it up is not for you to not read it, because it's too small, but think about the industries you're involved in. Where do those gaps exist? Do they exist right at the early stage, actually turning something into a usable innovation that's coming out of the lab, developing and refining it, or actually learning from the use of that product in the marketplace? I apologize, this is a slightly old version of this, but oh dear, never mind. Let me try and deal with this. So what are the lessons from COVID? It was a terrible pandemic, for sure. But there were some bright points that came out of it for the bioscience world. The governments of the world basically said, let's use the model that I described. Let's test things rather rapidly, give them conditional approval, and see if they work, and then give them full approval later. That's basically what we've done with the vaccines. But we'll also solve the reimbursement problem by telling you up front that this product is going to be paid for if it works. I mean, obviously, if it doesn't, you won't. And so it's actually been an accelerant for the whole bioscience world. And the key question now is, can the lessons that were learnt in COVID be applied more generally across the board? So let's finally think about European innovation. There's a bit of a problem. If you think about the comparison between Europe and the US, what's true about the US and its ability to innovate? Well, they do have some good universities, some good basic science, but what they do have is a very aggressive venture capital community willing to create a very large number of companies, allow them to go through Darwinian selection, and then they get bought by large American corporations, and all is then well. What happens in Europe? We have wonderful universities, we have great, bright innovators, but we don't have as much venture capital that is as adventurous as American venture capital is. And we also have very fragmented markets. So Estonia, 1 million people, the UK, 60 million people, France, 55 or whatever it is. So these do not represent home markets of substance in comparison to the US market. And so then each of those countries make their own purchasing decisions. I'm really wishing that I had run through these slides before, because they're not the ones that were finally sent. Anyway, what about how do we redesign this? Do we continue to play the same game in Europe that we play in the United States? Try and create large numbers of small companies, try and persuade investors to back them, and then try and see if large corporations, usually in America, will buy them out. What about this? What about creating European grand challenges, really grand challenges, that leverage the technology and science that there is in European universities, and back them enormously, providing they meet milestones? I mean, at the hundreds of millions. But then, importantly, as part of the deal, pre-agree that you're going to buy them across Europe, that fragmented market won't be there. Can we create a single market for European innovation? And can we do that with innovative companies that have pre-agreed relationships with large European companies, not just US companies or Chinese companies? So we really need to completely rethink, I think, the way in which Europe competes on the innovations stage on a worldwide basis. So I'd like to just ask the audience, since we have a minute or so, what would be the grand challenges that you would throw out to such a process? I mean, let me get you started. What about Europe creating the first zero-carbon autonomous flying taxi? I mean, that's one. I mean, another one would be, can you get an artificial intelligence program that redesigns and runs the healthcare system better than most people who do that? What would be, give us some ideas of grand challenges that might be fed into this model? Two of my colleagues are going like this to each other in the front, and the other saying, no, after you, after you. Grand challenges. Anything in the quantum computing field? Anybody here from quantum computing? At the back. How would you like to frame a grand challenge that would get hundreds of millions of euros behind what you're trying to do? Sorry? Annual supplies. Energy supplies, wonderful, thank you. My colleagues at the front here were generally useful in transmitting what you said. Yeah, so how could you apply quantum computing to optimize and improve energy supplies in Europe, which we all know is an enormous problem? I have 11 seconds for one grand challenge. And what technology would you start on?
That would be the challenge. And then
Okay, so grand challenge is the elimination of waiting times. The country that I come from, those waiting times are built and built and built. There's lots of people caught up in the system. How would you apply modern technology for that? So let me just finish with talking about my granddaughter. She is absolutely obsessed with these horses that have a horn sticking out of the forehead. You can't buy her a present without there being one of these horses with a horn sticking out of the forehead. So I've got to go back and tell her, you know, what was I doing? What was I doing was I was caught up in a herd of unicorns, young unicorns, larger unicorns, because that's how I see this audience and Estonia. I think it's fantastic, and thank you for having me.
Thank you, Professor Barker. Please stay on stage with us. One question from the audience.
Oh.
So, we've been asked, oftentimes scientists are not interested in the business. How could we, as entrepreneurs, help them to make their innovations to the market?
Oh, that's a big question, and I'm sure there's lots of people in the audience who have had experience of that. So, something was said in the first speech from this stage, right at the beginning of yesterday, which is the things that drive academics are different from the things that drive business people. It's getting grants, it's writing papers, and you have to recognize that is their first driving force. But you have to mix with them. You have to be sympathetic to the fact that isn't their main driver, that the commercialization is not their main driver. But you do ask these gentle questions. Okay, this is wonderful. You've created this CAR-T therapy. Isn't it fantastic? Now, who's actually going to use it? How much is it going to cost to get there? And what are some of the other problems and difficulties that we need to challenge ourselves with before we can say just thank you for the technology? So it's that kind of debate and dialogue. The practical problems, some of which I've alluded to here, which should be part of that discussion.
Thank you very much.
Thanks.
Big round of applause.